Thank you again. And not only for introducing me, but also introducing the subject as well. And I spent the last week trying to prepare this lecture, and I had to narrow it down a little bit as well because not only I wanted to show a lot of information, but also I wanted to show you what we can do to understand the impact of these parasites under one health. So basically, what I'm planning to do, sorry for a second. So how I'm going to split the presentation is, first of all, I will tell you who we are, what we are doing in my lab. We'll give you some background information on enteric parasites, and especially the parasites I'm going to talk about in this seminar, and then talk about how we use One Health approach to investigate Blastocystis and other gut parasites, but also what is the relation of these gut parasites and microbiome. And the microbiome is something we need to actually include when we are talking about zoonotic parasites and the effect of zoonotic parasite in health and disease, and of course under One Health approach. And then I'm going to talk about next big questions and projects we want to tackle as my lab as well. So who we are, for those that don't know, this is a UK, United Kingdom, England, Wales, and Scotland. We are in the southeast of England. This is Kent. It's also called the Gardens of England because the weather is much nicer than the rest of England. This is the University of Kent campus in Canterbury, and you can have a really nice view of the Canterbury Cathedral, which is the head of England's church. And yes, this is the Canterbury Cathedral. That's why the city is quite famous, but also about its research as well. My lab, this is a picture we had during the Christmas party. We're missing a few people from there, which I'm going to talk about later. The lab of my research is focused on investigations of the adaptations of microbial eukaryotic organisms and their course in parasitic evolution and diversity. So I'm going to look at not only the biological adaptation, but the environmental adaptations to see how these parasites are actually behaving and how they've been evolved. So we do a lot of genomics and transcriptomics, microbiome analysis, metabolomics. So we're looking at the metabolites that these parasites excrete, either in the environment or in their own. We do cell biology and biochemistry and aerobic biochemistry because these parasites live in a quite niche environment which lack of oxygen. That's why we focus on this anaerobic biochemistry. And the idea is to investigate the parasitic and free-living microbial eukaryotes living in diverse and extreme environments. And why I'm saying free-living, not only parasitic, is because understanding the close relative, free-living close relative of parasites, you can understand how these parasites have evolved. And it's important when you do investigations to look at both the ones that cause disease and not the ones non-causing disease and compare the two. Excuse me. So, which parasites we are working on? Mainly on the main focus of our lab is on Cryptosporidium, and this is a diarrheal causing parasite, and I will introduce it later as well. We work also in a non-gut parasite. This is Naegleria. Naegleria is quite famous for its ability to actually cause primary amoebic meningoencephalitis. So you need one of these organisms to go through your nasal cavities, goes in the brain. And this is also called brain-eating amoeba. And we want to investigate how it adapts in this environment, and if we can find ways to kill it as well. But also, another of my main organisms of interest is blastocystis, and we are going to talk about blastocystis quite a lot in this webinar, and why it's important. But as it's been mentioned, blastocystis is blastocystis is sorry, I'm going to list all the organisms that we are working on in my lab. In general, blastocystis, which is quite known, that has many subtypes. It has low specificity and question of pathogenicity. We are working on Cryptosporidium, which causes diarrhea in humans and livestock. And Amibia, this is another interesting organism, anaerobic as well, and cause amoebic dysentery. Giardia, another anaerobic, this is biloflagellate. You can see here, cause disease mainly humans and rarely some animals. We work with Eimeria, which cause coccidiosis, especially in chickens, but you find it in ruminants as well. And it can cause inappetence, weight loss, depression, and diarrhea, which can be hemorrhagic. And Prototheca, which is a non-photosynthetic green algae, infects cats, cattle, and humans. We recently actually found Prototheca to be in non-symptomatic humans in Thailand. But when it's actually present, it causes explosive diarrhea, which is a quite interesting organism to work with. I'm going to focus on two parasites because I'm going to talk a lot about this parasite during the webinar. And I will start with Cryptosporidium. Cryptosporidium is it's a single-cell epicellular eukaryotic parasite. We call it epicellular because it doesn't actually get into the epithelial cell when it infects, actually on the surface. This is the 4 different sporozoids that existed from the oocyst, and these move quite nicely to find a cell to infect. It's quite it causes cryptosporidiosis, and it's targeting quite a lot of the immunocompromised humans. There have been outbreaks of Cryptosporidium killing high population of HIV patients, but it's quite frequently found in children below 5, and in developing countries is the second major cause of death with children with diarrhea. So it is a quite important parasite. Even in UK, just to let you know that it's not only developing countries, it's everywhere. We have up to 6,000 people get infected, and this is the one we can monitor. And this is actually newspaper saying that it's been found in their water, and they are asking people to boil their water. Why it's of interest? There is no drug to deal with potential infection. There are more than 40 species that actually can be human pathogens. And Cryptosporidium is amongst one of the 5 pathogens that account the most of the 50% of diarrhea diseases in animals. So you can see that it's a quite interesting and important zoonotic pathogen. The life cycle is quite simple. We usually get ingested by the host. The oocyst is there ingested by the host. This is quite unique because the acidity of the gastrointestinal tract actually allows the cyst to break and release the oocyst. Actually, one of the reasons that people are asking to evacuate swimming pools when someone has diarrhea is because of Cryptosporidium. even the chlorine in the water of the swimming pool can actually make it the oocysts to break and release the sporozoites and make it more infectious. You can get the oocysts from either contamination water or by drinking or doing recreational activities. And they have the cattle there, but I actually found a much better life cycle, actually, which is more real, which is this. Usually the major source of the zoonotic parasite is from livestock and is mainly ruminants. So you actually get almost 90% of the animals will have or had exosporidium in their life cycle. So this goes in the water, this goes in the either by watering plants or by drinking this water, you get the infection in humans. I'm not going to go through the life cycle within the cell because it is quite complicated. We're still changing and we're still trying to understand it. And also it's not important for this webinar. The other parasite I'm going to talk about is blastocystis, which was discovered 110, 112 years ago by Alexei, and it was thought to actually be a yeast. And it looks, this is blastocystis, which what I'm showing here is different types of cells. And it's actually, it looks like a bubble. And a lot of misdiagnosis is because of this simplicity on the cellular level that blastocystis has. It was not until 1996 when Jeff Silberman actually managed to give blastocystis a home within stramenopiles. And I will come back to that. But it's quite weird that we have the position of these parasites there. Blastocystis has been found in the gut of individuals with and without gastrointestinal symptoms or diseases. And there's a big question whether blastocystis is pathogenic or not, or is an opportunist, meaning that it's actually goes be part of the microbiome, but it doesn't do anything. And the big question is whether the pathogenicity of blastocystis, whether one species can be pathogenic or not, could be species or subtype specific. This is another big question we want to tackle. But in the recent years, over the last 20 years, there have been several reports actually identifying blastocystis with gastrointestinal diseases, hearing diarrhea in children, with people with irritable bowel syndrome, IBS, again, IBS, IBD, irritable bowel and disaster, or another paper that actually found a single case of blastocystis. So why I'm showing that is the fact is that blastocystis, if blastocystis causes disease and so frequently, then everyone will have had, a lot of people will have had diarrhea all the time. And because it's been estimated that more than 1 billion people worldwide actually carry blastocystis in their gut. So the question is blastocystis an organism that causes disease or is there during a disease to actually help recovery, help people that actually have diarrhea to recover, excrete something or works with the gut microbiome to help recover from diarrhea and to become healthier. And this is something I will give you some examples of what's going on during my during the webinar. And this is something that really needs an One Health approach to tackle. Only by having an overview of everything that's surrounding not only blastocystis, but the host of blastocystis as well, will allow us to understand the biology and behavior of this controversial parasite. The life cycle, until recently, we had this life cycle, which we've seen that people ingest a fecal cyst of blastocystis from the environment, but I will come back to that later. I mentioned species, subtypes, and something that is quite interesting about Blastocystis is that there are at least 14 distinct subtypes has been described from birds and mammals, and this is through sequencing of the small subunit RNA gene. And why this is important and it's expanding is because we need to know whether certain subtypes, which could be species as well, can correlate with a disease or not. At least 12 of these were found in humans, 4 of which are more common. ST1, 2, 3, and 4 are the most commonly found in blastocystis. Subtypes in different hosts vary in their prevalence, and there are actually a different kind of clade of blastocystis in reptiles, amphibians, and insects, which is it will be a different webinar by itself. And all these kind of subtypes is the question how this is varies in the gene content. And again, if I have an opportunity in the future, I can talk more about the biology and genomics of blastocystis. I mentioned that certain subtypes are host-specific, and this is actually quite true. Subtypes in different hosts are very prevalent. For example, in ruminants, it's more frequently to have subtype 1. So the bigger the number you see there is the most prevalent subtype within the animal. So in ruminants, it's subtype 1. In humans, it's subtype 3. This is the most frequent subtype we found, followed by 1, 2, and 4. In chickens, for example, subtype 7 is the most frequent, followed by 6 and 4. And in non-human primates, subtype 1 is the most frequent, followed by 3 and 2. So in addition to that, just to make a little bit things more complicated, depending on the geographic distribution, we have different subtype distribution. What I mean, for example, subtype 4, we call it the European subtype, has higher distribution in Europe than in the rest of the world. So again, subtype 1 is subtype 3 is the most frequently found in both Europe and the rest of the world, but the second most in Europe is subtype 4. And we don't know what's the reason behind this and why and how important it is also. Another thing that is quite interesting is that recently we changed from this life cycle to this, and this is brought up by the CDC. And basically, they removed the environment from the life cycle of blastocystis, which I found it as a big mistake, and I will discuss this later as well. So what we wanted to do, I've shown you these 2 parasites, and I'm going to bring this map quite frequently throughout the webinar to actually discuss and talk about the different work we are doing, the different collaborations. Here's England, here is Somalia. We mentioned we are collaborating with tons of countries around the world, from North America to Africa, to Europe, and to Southeast Asia. And I will start with Thailand, and I will talk about the work we are doing with the Thai investigators. So we want to describe you the work on One Health in rural area, in tribes, school children, and in water. Of people as well. Sorry. And these are the people that actually have done the work on this. Okay, and this is a project we started 5 years ago, and this is by a PhD student called Vasana Jinatham. And she actually wanted to understand, explore blastocystis under one health umbrella using a rural community in northern Thailand as a model. And she actually went to the village of her parents to do this investigation. This is Thailand. This is the Chiang Rai province, is near Myanmar and Laos in the borders, and this is basically the community that she sampled from. As I mentioned, this is all the household we sampled from, and also we got samples from 45 people, from animals, and from soil and water. And this was to allow us to be able to investigate not only the prevalence of blastocystis, the presence of blastocystis, but also the transmission dynamics. So what we've done, we collected the samples. We've done cultures for all of them, and this is Vasana actually in front of all the cultures in Thailand, looking one by one at this tube on a daily basis for a couple of weeks. And she was not smiling by the end of this project. The cultures were looked under microscope, but we also do direct DNA extraction and molecular detection using both PCR and qPCR. We've done both due to financial constraints. Some of the work was done in Thailand, some of them was done in UK, and we were able to support this team to go through the whole project. When we start looking microscopically, we found that only, actually, that's in humans, only 4 samples were positive. When we did PCR, 12 of them were positive. When we did qPCR, 34. So you can see that the most developed the technique is, the better probably you have to detection these parasites properly. And all the samples were sequenced and checked to be correct that it's blastocystis. When we sequence them and we'll look at the subtypes, we've seen that almost within all the samples had 108 that were positive. 11 of them were subtype 1, 1 subtype 2, and so on. 37 were subtype 7, which is quite expected because most of them were chicken, and we know chicken has subtype 7. And we found subtype 23 in 12 humans, which was a huge surprise and making our story quite interesting. When we did phylogenetic analysis of this sample, we start looking at different patterns within that. So, for example, we've seen environmental samples clustering together, but also animal samples clustering with other water and soil. So you see chicken and water together, chicken here and soil. Water here, soil and humans. And humans here on its own, soil with humans, human and water. So how to interpret all this? Basically, we went per household and we wanted to investigate if there is truly zoonosis, if there is transmission between animals and humans, to make sure that what we are doing was actually correct and it's true. So household 1 had chicken. In animals, we didn't find any blastocystis in humans, and we have SD7 in the chickens. Household 2, subtype 1 and subtype 10 in humans, subtype 7 in the animals. So household 3, chickens again, subtype 23 and one unknown in humans, and ST7 in the animals. And this is what I'm going through the story, is that from all this, we couldn't see any actually transmission or correlation between the subtypes we found in humans versus the one found in animals. So the source of transmission is quite different. And this is the diagram. This is the diagram that's been used so far on blastocystis. What we actually found is that the water was one of the main source for both getting blastocystis or the soil. And this was the actually first investigation of blastocystis under One Health perspective, looking all the environments and the conditions of animals and people to see what's going on with the transmission of blastocystis. And this is the first paper that was published on blastocystis under One Health, and it came out in December 2021, and it was a pioneer study in the field. What we wanted to do after that is to investigate this case in other environments as well. So we managed to actually get ethic approvals to investigate blastocystis presence in tribe school children, again in the same area in northern Thailand. And also we sampled not only stools from the children themselves, but also from the environment, either from the water or the soil that children play there, because they spend a lot of time within the school as well. And one unique thing we've done, and another pioneer thing that was done, this was the first longitudinal study on blastocystis presence. So we didn't sample only at one time, we sampled 2 times 3 months apart to see whether actually there is colonization of blastocystis, or if this is something that is quite rare. So looking at blastocystis, we found 88% prevalence of blastocystis in humans, in children, 88% in chicken, 86% in pigs, and 71% in water. So most of the water provision of blastocystis in the school actually had blastocystis. When we wanted to look at the BMI scores, but also ethnicity versus blastocystis, and we've done all the relevant statistics of that, we were not able to actually identify any correlation between them. So blastocystis can affect anyone. It doesn't matter what kind of diet they have within this area. And this is actually makes stronger case that could blastocystis be, the source of blastocystis could be from the school itself. Looking at the longitudinal study, we found 2 cases where blastocystis is the same subtype maintained in 2 individuals, 3 cases, 2 cases was SD1, 3 cases SD2, 13 cases was SD3. So it remained. And in some cases, we actually had blastocystis coming in from absent to be present there. So we could see that blastocystis gets maintained and it can come as well. Similar to the previous report, we see under an One Health perspective, we can see clear transmission of blastocystis from animals and water to humans, but maybe the environment was actually played a much more important role in this. So these studies have revealed no clear pattern of direct transmission between human to human or animal to human in these communities. Instead, it points out the existence of multiple independent routes of transmission. Previous efforts investigating blastocyst sources of transmission have been geared towards looking at party relationship, for example, animal to human or environment to human, are misleading because we need to look the whole thing prospectively. Comparison of sequences found in the different hosts and environments indicate that highly similar strains of, for example, ST1 and ST3 are circulating in the communities. This suggests a shared blastocyst transmission cycle among humans, animals, and the environment for these subtypes. And additional communities, both rural and urban, should be explored under the One Health umbrella to determine whether similar patterns occur. And this is something that I really want to undertake with a project in Somalia as well. This paper was published in June this year in Epidemiology and Infection. And what we could see from both of these reports, we've seen a similar pattern occurring in circulation of transmission of subtype, but also a new player, a new kid on the block, which is the water. And Vasana, here, she managed to get a small grant from postgraduate studies in Thailand, and she decided by herself to go and do an investigation in the water, in the tap water in a community in northern Thailand. So she wanted to sample water from all these sources, and she actually did this work by herself, and then she contacted us with the results when she was ready. And what she's seen, and this was published last year in Parasites International, she's seen that most the samples, the sources that actually had chlorine as for decontamination had actually blastocystis. So this result prompted us to investigate how chlorine behaves, the concentration of chlorine behaves with blastocystis present. Sorry, how blastocystis behaving with chlorine, because it seems that chlorination doesn't kill blastocystis. So I had a very talented postdoc in my lab that had great experience in actually drug screening. This is Ruben Escolano. And I told him about this project, and he got super excited, and he designed it by himself and delivered it within a few months. Before I give you the results, I wanted to actually mention that according to the Centers for Disease and Control, CDC, the concentrations of 0.2 and 1 ppm, that range of chlorine are effective for eradicating both pathogens, and levels up to 5 ppm are considered safe in drinking water. So up to 5 ppm chlorine, we can actually drink it. More than that, it will cause us more damage. With that, looking into that, Ruben did a screening on blastocystis, and he actually seen that in some cases, in order to kill blastocystis, you need 200 times or more than this concentration, which this is undrinkable as well. When he actually exposed the parasites in chlorine, washed them, let them grow, and exposed them again, this went even 4 times more than it was before. So suggesting that they managed to deal with chlorine, and they be able to adapt and have resistance in chlorine and also hydrogen peroxide. So we've done some analysis, also microscopic analysis, to confirm this. Red here looks the activity of of blastocystis, um, showing that they are alive because it's targeting mitochondria, basically. And after chlorine, you can see that there is still activity in some of them, so they can actually resist. And this is with IC50, which is supposed to kill half of them. So we demonstrated that all blastocystis isolated included were highly resistant to chlorine, including concentrations ranging from 175 ppm to higher than 1,800 ppm to eliminate any chance of recovery. The biochemical and cell biological results herein suggest that other water treatment processes, either chemical or physical, should be applied to eliminate blastocystis in the water if we want to eliminate it. For example, pre-chlorination treatment stages such as sedimentation, coagulation, fluctuation, infiltration should be used in the water disinfection procedure. And in rural areas where it is often not possible to include these necessary treatment stages, blastocystis remains in the water, maintaining transmission cycles, and we should expect having blastocystis in the sample. And again, this is how I mentioned and insist not only to when I give talks and to my students, but also all around, that we need to look always a holistic approach to investigate what is going on and understand how an organism is behaving. And moving from the natural environment, outside environment, I'm going to the environment in the gut. So just an introduction on gut microbiome. I hope you are fully aware of what I mean by gut microbiome, but I wanted to remind you that are the microorganisms, including bacteria, archaea, fungi, protists, and viruses that live in the digestive tract of animals, including us. And the Caafimaad mutation is the aggregate of all genomes of the gut microbiome. So I wanted to remind you this because I'm going to mention this quite frequently. So in the last decade, there have been a couple of reports, a few reports on investigate blastocystis relation with microbiome, and they found that carriers have higher bacterial richness when carriers of blastocystis have bacterial richness, which means a more diverse microbiome, which means a much healthier microbiome. And this is healthier carriers again here have blastocystis. Carriers have higher bacterial diversity. Carriers have lower bacteroides and higher bacterial niches, and carriers have absence of gastrointestinal activities and inflammation. And a report that was actually published in Nature Medicine a couple of years ago actually emphasized that microbes such as Prevotella and blastocystis were indicators of favor postprandial glucose metabolism, and basically suggesting that healthier individuals had blastocystis and Prevotella. Okay, so again, another case suggesting that blastocystis actually could be an indicator of a healthy microbiome. On the other hand, reports of certain blastocystis like SD7 here in vitro actually look at that can actually cause some pathogenicity and some changes in the gut microbiome. So there are positive sides of certain subtypes and negative sides of others. So what we started doing, and this a work that was initiated by Jamie Newton here, a PhD student that is actually writing up his PhD as we speak. We wanted to look how the metabolome is changing according to the presence of blastocystis. So I contacted 3 PIs around the world, Fundan Dokurman in Turkey, Jong-won in South Korea, and Elenike Didekagi in Thailand. And I told them, why don't you, when you look at blastocyst positive samples, can you please extract the metabolites from the stools to see if there's any correlation with the presence of blastocysts? So we've done animal metabolomics, which is a much faster and more stable quantitative stage of measuring metabolites in the stool samples. And we compare these. Colorful is the positive, black is the negative sample, and we've seen significant differences between the different stages. So, as I mentioned, Thailand, Turkey, and South Korea, blastocyst presence absence, we actually have distinct metabolic profiles. What that means, either this is a favorable condition for blastocysts to grow, this metabolic stage, or blastocysts secreting or help the secretion of different metabolites to make, to be able to recover and have a different metabolic profile, which means healthy microbiome. And we suggested also certain metabolic pathways that could actually help, and this is actually correlated with intestinal inflammation, so which helps abiosis, so it makes much healthier. So blastocysts might not be really a pathogen and a parasite, but it could be an organism that could help and support healthy microbiome. So having this report out, Jamie decided to expand this in Southeast England and look at the cohort here in the Kent area to investigate humans with or without blastocysts. So this was in a really bad time as well, because as soon as we started, COVID hit. So we were not able to actually sample the numbers that we wanted, but with the samples we managed to get, we actually identified that most of them had subtype 3 followed by subtype 1 and 2, and then last was subtype 4. So the presence of different subtypes amongst the people we investigated follows the pattern of the rest of the world than Europe. We collected all the metadata. Again, another One Health approach to look at everything from gender, age, whether they take probiotics or vitamins, if they have pets, whether they have gastrointestinal symptoms, whether they were drinking milk, whether they were vegetarians or vegans, and so on. And then what we've done, we look at the microbiome profile of all these positive samples. And what we actually saw is that depending on the subtype, there was no significant difference between the microbial profiles. And looking at the metabolome, so we extracted the metabolites as well with the gut, we actually had a quite nice overlap. So, and people with or without diseases were not separated in all these cases. We look at different important metabolites that could be correlated with potential disease, and we've seen that in blastocyst presence absence did not change significantly their abundance. So what is the take-home message from this project is that this is the first study to focus on the epidemiology of blastocysts in this part of England. And we also investigate the impact of the gut microbiome and metabolome and its role in irritable bowel syndrome and ulcerative colitis, which are induced by dysbiosis. We found high prevalence blastocysts, with STs to be the most common. IBS and colitis impacted both the gut microbiome and metabolome, regardless of blastocyst colonization. but we suggest that further studies with larger cohort involving the serum and urine metabolome, as well as the fecal metabolome, and in vitro studies, including histopathological examination, could be largely beneficial to the overall understanding of the impact of blastocysts on the gut microbiome and its role in this biosis and in health and disease in general. We did not find a significant difference in microbial diversity in relation to blastocyst subtype or blastocyst colonization in combination with impact factors. However, studies with larger cohorts are indeed needed to be understand these factors. So I spent the last half an hour or so talking about blastocystis. How about other parasites? And I introduced a few of them in the beginning. So now we need to change a little bit more and move from Thailand to Bangladesh, where we actually look at rural areas to investigate the presence and prevalence, presence, and transmission dynamics of different parasites. And this is a work that is being done by a brilliant PhD student in my lab, Sadia Maxamud, which is shown here. And Sadia, unfortunately, she started also during the COVID period, so we were not able to go and sample ourselves. So we need to establish new collaborations to do that. So Sadia looked at more than 200 samples that were sent to her by the collaborator, Professor Caafimaad, in the Agricultural University in Bangladesh, where she sampled the area I showed you before, the area here, which is a rural area in Myanmar. And basically, she identified rarely the presence of blastocystis in humans. There was, she found only one case of blastocystis being infecting humans, and one case of Cryptosporidium. But she also identified that Cryptosporidium and Giardia can be found in lots of animals. And this is a summary of a table with the animals infected with a co-infection of different parasites within the same host. So what was nice to see from this project was also that we couldn't see any correlation between blastocystis and Cryptosporidium in humans and animals, which was quite interesting. The strongest part was we could actually see one subtype blastocystis tend to be circulating between goats, chickens, and pigeons for some reason, which we are investigating this even further. The detection of diverse Cryptosporidium blastocystis species in the study has provided valuable insights, though, into their overall presence and prevalence, not only in humans, but also in a variety of animals and soil in Bangladesh. This, again, underscores the significance of adopting a One Health approach, recognizing the interconnectedness of human, animal, and environmental health for a comprehensive understanding and management of intestinal parasitic diseases. And continuous investigation are important to enhance our knowledge of intestinal parasite transmission, especially in developing countries. And moreover, conducted future One Health studies in Bangladesh, encompassing larger populations, and expanded the research scope to include various study areas and studies such as different water sources will give valuable insight into intestinal parasites and their transmission dynamics. And we are currently writing, hopefully, something in this project in a couple of weeks' time. Following that, we are moving continents, and we are going to Algeria, where we actually undertake several projects to investigate transmission and the presence of parasites in various animals. For example, this is a paper that was published last year, where we look at Cryptosporidium in farm marine and freshwater fish in different areas in Algeria, and we identified that 5% of the fish actually carry Cryptosporidium, and this is one important thing to actually investigate and properly cook the fish before we eat it. We look at Cryptosporidium giardia in camels from Algeria, and we look at 2 different areas. And why it's important to look at camels in this area? First of all, it's been used for many reasons, including harvesting of milk and drinking milk. The camel milk is important in this. Drinking camel milk is very important in these civilizations, and we wanted to make sure that there is no pathogens carried. And we look at different characteristics, including sources, ages, and breed. And we found that in some cases, in certain areas, we have circulation of Cryptosporidium and giardia within camels, making this very, very important, and investigate this even further. And as soon as we've observed that, we decided we talked with our collaborators to actually collect samples in other areas and look under One Health perspective. So they collected samples from households where they actually had animals living there as well, and collected soil, water, stool samples from humans and the animals to investigate the potential transmission dynamics. And I know you are not able to read all this, but this is a different household we are actually investigating, and this is the presence of parasites that we identified in this household. And I will give you just one example. This is one household where we have goats and we have sheep. We have sheep and cattle within, and we identified Cryptosporidium circulating, mainly Cryptosporidium bovis, giardia circulating with these animals, blastocystis quite a lot, and blastocystis SD10 to be present. So here we can see that blastocystis could be circulating not only within the animals, but also to humans as well. And there is a pattern of transmission dynamics. In another case, we've seen blastocystis SD1 be present in the humans, but in versus SD1 and SD2, sorry, in the humans, but in the animals, there's only blastocystis SD7, and giardia is only in the animals, it's only in the cattle. So there is no transmission between the animals to humans, or vice versa in this case. So again, we put a whole big story on the circulation of blastocystis under One Health in Thailand, and we come to Algeria and we've seen this pattern breaking. So maybe there is kind of different environment that supports the circulation of these various subtypes or different species of gut parasite within that. We talk about Cryptosporidium and the transmission of Cryptosporidium is a Cryptosporidium species. I want to actually kind of finish the focus on understanding the role of parasites in the gut by actually talking about an in vivo and in vitro study we've done on Cryptosporidium. So now we are moving to France, where we've done the in vivo study, and in Czech Republic, which is here, where we've done the in vitro study. So before talking about the study, just some background information. So in cattle, we've seen that depending on whether there is a normal delivery or cesarean section, we have different types of microbiome being developed with from a few days up to 2 years old of the animal. And what they actually demonstrated, this is a nice review on the gut microbiome and development of the gut microbiome in cattle. They show that they suggest that maternal influence, whether the delivery and what the mother is eating, could influence the microbiome of the young calves. The diet, whether it's liquid or solid, the age change the microbiome, weaning, basically what kind of food is eating, the use of antibiotics and food supplements also helps the gut microbiome. What they forgot to mention, they haven't mentioned, this is where our research is becoming important, is the influence of parasites like Cryptosporidium. And as I mentioned earlier in this talk, is that almost 90% of the animals at some stage will get Cryptosporidium. So this will actually change the microbiome. And so we developed projects to characterize the impact of Cryptosporidium parvo infection on the composition of the gut microbiome, and try to provide further insight in the functional structure of the microbiome in response to parasite infection, but also assess the collective change of metabolites in the gut during Cryptosporidium infection. So we've done 2 studies. We've done a blind study. This is the in vivo work. We went in the field and collected stool samples from dairy calf aged between 7 to 21 days. And the Petro, which was a PhD student working on this, was actually going on a daily basis collecting, and in the evening was doing the DNA extraction and the metabolite extraction of each of the samples. We collected 134 animals, and we divided without Cryptosporidium and with Cryptosporidium. And then we split them in this further. 75 of these had Cryptosporidium but no diarrhea. 39 had Cryptosporidium and diarrhea, and 20 diarrhea and 4 Cryptosporidium. So we also quantified the presence of Tsaousis in the sample using qPCR. This is Petro that has done most of this work. Another student that is finishing now his PhD. And we actually seen that depending on whether there is Cryptosporidium and diarrhea, non-diarrhea but Cryptosporidium, and non-diarrhea and non-Cryptosporidium is this one. So this is kind of the healthy control. We've seen kind of different patterns changing. Depending on, this is a control, this is a higher enriched groups of different bacteria. For example, if there is no Cryptosporidium and no diarrhea, we have high prevalence of Bifidobacterium, which is one of the groups of organisms that are indicated of health microbiome. Where we have diarrhea, we have Fusobacterium, which is an indicator of a disease. When we've done a random statistical analysis, we actually found that certain bacteria groups actually are more correlated with certain disease status in correlation with Cryptosporidium. As we've done with the human blastocysts, we've done in the cattle with Cryptosporidium, we've done NMR analysis, and we've seen that certain metabolites are actually changing in relation with the presence of Cryptosporidium. We've seen acetate, lactate, and butyrates to be highly abundant when you compare diarrhea versus healthy samples. And statistical analysis shows clustering of certain metabolites and depending on the disease. So depending whether the microbiome is actually friendly to Cryptosporidium. It doesn't cause diarrhea. We have a different pattern than the ones that cause diarrhea as well. And now we are investigating this even further to see what kind of indicator we can actually get to show actually presence of Cryptosporidium and disease and how we can solve that. And this was an in vivo study. This was in the field. So getting this actually results prompted us to actually do a study of our own to take young calves and infect them with Cryptosporidium and monitor on a daily basis because it's happening in the microbiome and metabolome. So Pedro went to Czech Republic. He got 2 groups, 5 calves per group, infection with Cryptosporidium parvum, non-infected neonatal calves, and we've done sample collection every day from 0 to 15 days, and then every 3 days after 21 days. So we got 170 samples to analyze. How we've done the work, we start with infection at day 0, sample collection, daily assessment of calves. So we've done health assessment following Riggs and Schaefer model. We look at oocyst shedding using qPCR and airline carbomethyl violet microscopy. We've done microbiome profile with 16 RNA acceleration sequencing, and we've done metabolomic analysis with NMR metabolomics. So this is the 2 groups of organisms, the healthy ones, and this is the infected ones. And this is actually Pedro looking at the cows. And this is, of course, this is the healthy cows and monitor them, the health status and temperatures and so on. So as I mentioned, collected samples on a daily basis, did qPCR and PCR to look at subtypes, microbiome sequencing, and metabolomics. So I'm giving you an example here of what a healthy microbiome look like. This is day 0 and going to day 21. We have Clostridia here. This is a bacterium, which is the orange one, which is correlated with a healthy microbiome. The dip here you see in DM10 is when they change from a liquid food to a solid food. So we have a little bit down, but Bifidobacterium is recovering. And we have Fusobacterium very low in here, and Lactobacillus, which is this color there. So this is what we can see as a healthy microbiome. The fecal score, this depends on whether there is diarrhea or not, is constant. So there was no change, there is no diarrhea. Gain weight here, and the temperature of the cows is stable, as you can see here. So try to memorize this, and let's go to an animal that is infected. So we infected at day 0. The animal, it takes 3 to 4 days to start for the parasite to start replicating and increasing. So after day 5, we can see higher abundance of oocysts per gram. And then it's reducing after day 16. And the fecal score, after day 5, we start looking at a more liquid kind of feces. The weight of the cow kind of went a little bit straight and increased near the end. And the temperature were 1 to 2 degrees higher than in the control. And how the microbiome look like, this is how microbiome look like. Bifidobacterium kind of reduced quite a lot, trying to increase at some point to help with the recovery of the diarrhea, but completely decreased by day 7 here. Clostridia start identify and increase, Fusobacterium, huge prevalence of Fusobacterium with diarrhea, which shows correlation of blastocystis, cryptosporidium with Fusobacterium, and lactobacillus only recovering near the end. So we can see two different patterns found identified in these cases. And how about weight? This is infected animals become more stable during the high diarrhea cases, which is expected where there is a more linear correlation in cows who are not infected. And as I started this part of the presentation, and I mentioned, we need a holistic approach to monitor how an animal is developing, but we also need to look the parasites, how the parasites influence this kind of behavior in the gut microbiome and in the health and disease of the animals. So future studies, currently I have a PhD student that started this year. Yasin Al-Saman is working in Iraq, collecting samples to look at the correlation between parasitic disease in humans, animals, and the environment. But not only gastrointestinal parasites, but we actually look at tick-borne diseases as well. And this is with Malamud and Dalia, two excellent parasitologists in Iraq supporting Yasin for these studies. We have projects in Turkey, and this is ILM Ankur and Funda Dogruman, which are collecting samples they're doing one clinical study in this area in Turkey, and it's kind of interesting, this area, because it actually changes depending on where between autumn and spring. In autumn is quite dry, and people are living in this area. These people are here. You can see in April, May, there is a big dam there as well. So we want to look at the transmission, circulation of parasites during the two different time periods as well. Last but not least, we are doing a study in Cyprus, and this is another PhD student, Dr. Daphne Mavridis. She's looking at two different projects. One to look at a holistic One Health approach on transmission of parasites, gastrointestinal parasites in farms. And we're going to look at the environment, the pests around, and the farmers. But also we initiate a study to look at the environmental impact, the pollution had to do with the animal health and human health and the transmission dynamics of parasites. So future work of my lab is to understand the evolution and adaptation of neglected gut microbial parasites and their role in both health and disease. Explore alternative non-pharmaceutical means to prevent zoonotic gastrointestinal parasitic disease that pose an epidemic or pandemic risk. And overall, to improve human, animal, and ecosystem health by integrating metadata related to human, animal nutrition, health status, and overall biology. And as I said several times, we need to look environmental, ecology, veterinary, public health, human medicine, microbiology, economics, from individual health to population health to ecosystem health, and everything under one health, to look at the zoonotic infection of these parasites. And with that, I want to thank everyone, all my collaborators, especially my lab members, Sadia Mahmoud, Petro Pinto, and Jamie Newton, and Ruben Escolano, which has done most of the work I presented today. My huge support by Dr. Eleni Kendekaki, which actually helped me in most of this work, not only in Thailand, but in other projects around the world. And now Eleni moved from Thailand to Cyprus to support Daphne, but she got the position at the veterinary institute there to continue that. In Bangladesh, MD Shadi Suman, in Algeria, Karim and Nasipa, in Iraq, Maulud and Dalia, in Turkey, Ailem and Funda, in Cyprus, Daphne, and the funding bodies from Thailand to University of Kent to Enteric project and Microbiology Society. And I want to actually mention that we are running a Blastocysts under one health project. This is a network project that we want people to investigate blastocysts under One Health, and we want your collaboration as well. And I hope at some point we initiate that with the university there to do a similar project in Somalia, and we want to explore this even further. And I will leave you with that, and I'm open to any questions. Thank you very much.
Thank you, Dr. Anastasia, for your nice presentation. And you also shared us the work you are doing. It's really amazing. Thank you. We are very happy to have you here.
Thank you.
Thank
you very much. Do you want me to stop sharing?
Yes, you can. You can stop, please.
Yeah. Okay. Sorry.
Yes, nice.
Okay.
Dear participants, please feel free to ask Dr. Anastasia if you have any question. Feel free to ask, or otherwise you can write in the chat box so we can address.
Okay, I like that. I will start with the question that Mukhtar Ali actually put on there. Yes, there are limited resources, and I understand that. To be honest with you, the rapid antigen test is not very accurate anyway. It's expensive as well. I don't even use it for my research because it's, as I said, it's not accurate and it's quite expensive. I wouldn't use it to look at crypto. What I would suggest, there is training of people, even with limited resources. I think the molecular diagnosis is becoming very, very cheap. I actually use a company that provides tag polymerase at 1/5 of the price that normal ones, and it's actually is very accurate anyway. So, and what I would suggest is organizing programs to get people in the field trained, either coming to Europe or bringing experts from Europe to come to Africa and help you train to do this by yourself. Because I think you have the experience, you have the expertise, you have a really good quality of scientists in Africa that can actually do this. So, and at the end of the day, I think with limited resources, you can actually identify, find ways to overcome the expensiveness, let's say, of some of the issues, and you can do the diagnosis yourself. There is a great initiative on genomics in Africa, which has been run quite successfully recently. And I've been following a lot of initiatives in Africa, and there is huge financial support. I don't know where the money is going at anyway, but I mentioned to people from Somalia many times, if you want any help or any support, I'm here to help you to run projects together. Any other questions?
I think for now, no. I would like to invite Prof. Dr. Professor Abdullah from Abrar University. He's a parasitologist. So if you would like to say something, please feel free for Abdullah.
Hello, everybody.
Yes.
Hello.
Hi.
Okay. Good morning there, and good afternoon, and good evening somewhere. Thank you so much, Professor Anastasios. I'm very glad to listen to you for the second time. I think yes, before I think yes, I did another presentation. I think I was just like a student here, particularly with the issue of blastocysts. Blastocyst is very new things, and I like everything said about that, particularly the story. You are taking the parasite as a story from beginning up to the end. Just really, my only comment is to say thank you for being here, and thank you for this presentation. And I am sure everybody is following the film of enteric protozoa parasites here. And we hope really we can do something, or we can continue collaborating with you in Africa here, or in Somalia in particular. But it is very interesting, and thank you, everyone. Thanks.
Thank you.
Thank you from Dr. Nice words. Any questions so far, please feel free.
As I mentioned, if you have any questions as well, you have my email, you have my Twitter account, please to contact me. It has been recorded. So if you want to listen to this again and you come up with any questions, feel free to contact me. I would love to collaborate with you guys. I would love to come to Somalia as well. Sadia Mahmoud is actually, she's Somali, and we were trying to actually get her there, but we couldn't. Hopefully we'll find another opportunity to do this. And I'm here to support you however I can. So if there are any initiatives that you want to apply for, or if you want support with grant applications, whether are locals or international, please feel to contact me. I see that, especially in African continent, I'm not going to say only Somalia, I think it's a whole African continent. The research has been significantly neglected, and this is I don't know the reason behind that. It's probably the Western civilization that is doing that, and this is really bad in general for the community, for the research as well. So bring your dreams, bring your research, and let me know if you need any support. And you need to be more provocative. You need to start looking into the research. You have a huge biodiversity there that is unexplored, and you guys need to do it. Don't bring anybody from outside. You should be doing this because you have the abilities, and there are several people like me that wants to bring support to African countries. Let us know, and we can help you as much as we can.
Thank you. Thank you for nice words and advices. We really appreciate. For just a reminder, we will post this recording. It's already recorded, so we will post on our YouTube, so you will be able whenever you want. I think we have one hand raised. Please feel free to ask.
Hello, everyone. Good afternoon, good morning, everyone. And I wanted to ask you one question, the professor, about this parasite, Cryptosporidium. How about the geographical distribution? Is there any limited factors that are restricted the distribution, the geographic distribution of the parasite, either in Africa or Somalia or in Asia or other areas? How about the geographical distribution and how they affect their transmission? Okay, thank you.
I don't think there is any. First of all, in Somalia, I'm not sure. This is a project we were trying to actually put together, and there some of you are here that collaborated with me. We tried to collect samples to do this, but unfortunately we're not able to actually export these samples from Somalia to check. But from research we've done all over the world, or research has been done from the Gates Foundation, basically there is no constraints. It's everywhere, and especially if the transmission is typically coming from ruminants, from calves, goats, and sheep. So if you have them nearby, you probably can get Cryptosporidium in humans. In animals, I'm pretty sure 80% to 90% of the animals will have it. In big cities, it's quite rare, but there have been occasions, even in the US, where Cryptosporidium was actually in the water supply, and it killed 100 people that were immunocompromised when people started drinking the water. So I don't find any reason to be restricted at any point the prevalence of Cryptosporidium. Certain species and certain subtypes could be have a geographical distribution, but this area is quite unexplored, and it needs further investigation.
Thank you, Professor. Before we conclude, I would like to remind you all. I want to remind you all about an exciting opportunity on the horizon. Our upcoming One Health postgraduate certificate program promises a further enrich your expertise in this critical field. Stay tuned for more detail and be ready to embark on the next chapter of our One Health journey. It's still open. You can register with an open hands. Thank you for your dedication, engagement, and commitment to the well-being of our community. Together, we can make a lasting impact on public health, and I look forward to our continued collaboration in the future. Thank you, Professor. Thank you all.
Thank you very much. Have a nice day. Thank you for listening.
You too.
Bye-bye.